what is innovation and research, R&D? I came across this famous quote from Dr. Nichestund, who was vice president of 3M. He said, research and development is a transformation of money into knowledge. So you have money, you do research and development, and you generate a knowledge database, right? And then use this knowledge through innovation and make it commercially viable, right? Convert this idea or this knowledge into something that can help the patients, or in the context of pharmaceutical, that helps the patients, right? So that gives you the commercial value. So that's where, R&D and innovation, they are interlinked, and it's really a cycle that you have money, invest in R&D, generate a knowledge, use that knowledge, do innovation, generate more money, and again, invest that money into R&D. And that's how the cycle continues. So let's look at pharmaceuticals, right? And maybe I am wrong, but my understanding is, at least at the academic level and in general, all of us just think that, okay, pharmaceuticals are like, just a small molecule or the chemical drugs that we see on the shelf on the medical store or the pharmacy. Like we know paracetamol and those kind of drugs, all the chemical drugs. But then there is a lot more to it. In the last couple of decades, it has changed dramatically. There has been a significant progress. And now you see medicines, as small as aspirin with molecular weight of 180. Then you have hormones, peptides, antibodies, all the way having a molecular weight up to 150,000 Dalton. So it's like, it's a huge, I would say, the progress from a smaller molecules to the large molecules. And day by day, I see the global pharmaceutical trend is shifting from small molecules or the chemicals to more biologics, proteins, antibodies, and those kind of things. So in my next slide, this slide, and these are just few examples. I'm not saying this is an exhaustive list. I'm not saying this covers everything. But these are some of the drugs or some of the modalities, we call it, different pharmaceuticals that are currently in development in the market, and globally, a lot of research is being done on these modalities. This is something we all are familiar, like the small chemical molecules. There are a lot of peptides. All of you must have heard about Ozempic, which is like a largest selling drug right now globally, for weight loss and the diabetes treatment, semaglutide. And what is that? That's a peptide. And, these are slightly bigger than small molecules, but then they are not as big as the proteins. They are peptides. It's a very important class of drugs. Then there are a lot of proteins. You have enzymes, a lot of enzymes for therapeutic applications. Interferons, probably this we study in the syllabus, the interferons. A lot of cytokines are being developed for treatment. Hormones are being developed. GCSF is a classical example, which is already there in the market. But then there are a lot of other hormones that are being developed. Radioligand therapy, this is a very interesting modality. We know that for the cancer patients, they go for a radiation therapy. And when you give external beam radiation, we call it. And when you give radiation, say example, you are targeting a lung cancer, but you're giving a radiation, it damages a lot of healthy tissues as well. And that's where we see a lot of side effects, toxicities, etc. But there is a new development in the science where we develop a vector or a targeting molecule like an antibody, and then attach it to the radioisotope, and that goes and specifically delivers that radioisotope to the particular cancer cell. So the effect of the radiation is only on the cancer cell. It does not kill the healthy cells or the neighboring cells. So this is a huge progress that we are making. On the similar grounds, there is another one called antibody drug conjugate. The one difference between radioligand therapies and antibody drug conjugates is, instead of radioisotope, the antibody here is attached to the cytotoxic payload. We know the chemotherapy for cancer, there are a lot of drugs which are cytotoxic. They kill the cancer cell. But they also have a lot of side effects because they also end up killing the healthy cells. And to make it more specific, again, this is another modality, antibody drug conjugate, where the antibody takes the cytotoxic drug or the cancer therapeutic small molecule to a particular cancer cell, delivers it there, and exerts its action only on that cell. So you minimize the side effects, because you must have seen what cancer patients go through when they go through a chemotherapy treatment, right? The monoclonal antibodies, this is another very, very important class of molecules. These are, there are multiple formats for these monoclonal antibodies that are there for immunology, cancer, and a lot of other indications in the market. The cell and gene therapy, these are two classes which are like very, very innovative. CAR T cell therapy, here, you are collecting the cells from the patient, genetically engineering those cells and putting them back into the patient, and then those are going and killing the cancer cells. So this is a very innovative way. There is a gene therapy where you are manipulating at a gene level, to have an effective treatment. And again, there are more examples like oncolytic viruses. There are vaccines. Vaccines we only know as a preventive vaccines, right? But there are also therapeutic vaccines. So there is a lot that's happening right now, and each of these topic can have a symposium in itself. It's such a vast and complex topic. But I think, to keep it simple in the beginning, I think today I'm going to focus more on the small molecule, because that's what we are more familiar with, and then probably later on, in future, if there is more interest, we can talk about other topics. But right now, today's talk, I will focus mainly on the small molecules. And if you look at, in general, the value chain for the pharmaceutical research, we know that India is a powerhouse, or the pharmacy of the world, we call it. We supply a huge proportion of the global generic drugs in US, Europe, and everywhere else in the world. And Indian pharmaceutical companies have been mainly focused on the generics. Of course, there are few which are focused on drug discovery and development, but our major focus has been on the generic so far. And I'm hoping the trend changes now. And there are multiple generics, super generics, or the generic plus, or the complex generics, we call them. And then there is also a biosimilar. So for the small chemical molecule, you have generic drugs. For large molecule, you have a biosimilars. And then there is a novel chemical entity, NCE, or if it is a biological molecule like protein or antibody, you call them novel biological entity, right? So it's NCE or NBE. But this is essentially the drug discovery and development outcome, that you come up with a novel molecule. So as you go up the value chain, the complexity, the investment, the resources, the time, and the risk increases tremendously. But the return on investment also increases proportionately, or I would say many fold. So you spend a lot of money, but then if you are successful in getting one molecule out in the market, I think that changes the fortune of the company. So I think this is a high value, high risk game. But I think end of the day, what it does is it really helps the patients, because patients get access to the new medicines. That's most important. I don't want to explain this slide, but just want to tell you, this is a slide to give you a glimpse of how complex the drug discovery process is. And I'm sure the speakers who spoke for last few days before me, they must have covered on the drug discovery aspect. But it's an extremely complex process which involves hundreds of scientists, different expertise, different functions. So yeah, this is just to indicate the complexity of the drug discovery process. I don't want to go through everything. And probably you might have seen this in the previous presentations, but then I just want to spend a minute here. So if you look at the small molecule drug discovery, and we start with like 20,000, 30,000 molecules in the beginning, and then it goes through multiple steps, multiple processes of lead optimization, discovery screening, then ADMET profiling, that the DMPK and toxicology profiling, then they do clinical trials. And finally, only one drug reaches the market, right? And how much time does this take? 10 to 15 years. It's a long game. And how much money do you need? 1 billion to 2 billion US dollars. And just to give you a sense of what 1 billion USD means, given the current exchange rate is 8,900 crore for $1 billion. So it takes somewhere around 9,000 to 20,000 crores for one drug to come to the market. And obviously, the pockets need to be deep for the pharmaceutical companies, and we need to really have that kind of a patience, commitment, and the resources to be able to wait for 15 years to get the result, to get the commercial benefit of the research that we are doing. And that's the reason, not all the companies can afford to do this drug discovery. And I have seen that a typical model is Indian companies do a research to a particular stage, and then probably they out-license it to the bigger pharmaceutical companies globally for further development. Just wanted to give you a sense of what this game is about, how much does it cost, how long does it take.
Toxicity, sir.
Yeah, please go ahead.
Yeah. Yeah. Out of these 5 steps, which is the step where maximum cost is involved?
The clinical development, clinical trials, and particularly this is I will come to that in the subsequent slide. But the clinical trials are the most expensive ones. They are really, really expensive clinical trials. But I will cover it a little bit here. Yeah. So if you look at this process now here, okay, this covers the entire drug discovery process. So you start with identifying a target. When I say target, this target is like, it can be a receptor, it can be an enzyme, it can be hormone, it can be ion channel, anything, right? Where you expect your drug to go and bind. We study, right, G couple protein receptors, etc. So your drug goes and binds to that target. You identify that target, understand if that target is druggable or not, and understand if that target is associated with some critical functions, biological functions or not. Then you identify compounds, then you start screening multiple compounds, the library of compounds against that target, whether or not your compound binds to that target. It's important that your compound binds to that target, then only you can see the pharmacological action. And then whether it is agonist or antagonist. You establish activity in vitro, and then you also do some animal models of disease to generate some pharmacology data to understand the activity. I'm talking very high level. Each of these has a lot of complexity and sub activities, but then I'm talking very high level. And then once from multiple thousands of compounds, you arrive at like say 10, 15, 20 compounds, you screen them further, then you choose a couple of candidates that, okay, these are the drugs, these are the candidates or the chemical molecules that seem very promising and we can take them forward. And then that's where you do the preclinical testing or the drug safety testing, right? So you have a candidate here. This is called a discovery stage. And once you identify a candidate that can go forward for the development, then that's called a development stage. And this is where your preclinical safety, non-clinical safety, or the toxicology studies come into picture majoritarily. You have to do all those studies. And then there are 2 places where there is an important regulatory approval that you need before you put it to phase 1 trial. That's, we call it first in human trial, FIH trial. So from animals data, based on animal data, you propose that we go to humans now. And what does animal data compromise? The safety and efficacy. And based on that data, you propose that we want to go to humans, but you need to go to regulatory agencies like FDA or in India like DCGI, and then get an approval that this is the data, this drug is going to be safe in human. Based on that data, agencies will approve whether or not you can go to human. Then you go to human. There are 3 different phases of trials for development: phase 1, phase 2, and phase 3. Phase 1, you take limited number of healthy volunteers, usually. Only for cancer or oncology, you take it in patients, but otherwise you do it in healthy volunteers. You want to determine the PK, pharmacokinetics, and safety in those subjects, but limited number. Then once you establish that, okay, it is safe up to this dose and it is good, then you go to phase 2. This is very important trial. In phase 2, you study the efficacy and safety, but mainly efficacy, proof of concept that, okay, you wanted your drug to work on a diabetes or a cancer or inflammation, and then you really check here in the patients whether or not it's working. This is a phase 2 trial. This is very, very important trial. A lot of drugs fail here in phase 2. And then everything is done. This will be like, say, example, 500 patients. This is phase 1 is like, say, 40, 50 kind of healthy volunteers. Phase 2 is usually in hundreds, 500 patients, example. And you go to phase 3 trial. This is a long-term multi-center, like globally, because your drug is going to be given to US citizens, Europeans, Chinese, Indians, Japanese, and then different genetic things, and then all those things also come into picture because different people from different countries, different origin, have a different genetic constitution. So you need to test it on all the population. Wherever you want to market it, you want to market it in Japan, you need to test it in Japanese patients as well. In India, then Indian patients as well. So you do a global multicentric trial, and then you do the safety and efficacy, the phase 3 trial. And this is the most expensive one, followed by this one, and then this one, and then it goes down. But then phase 3 trial is really very expensive. And then once you have all this data up to phase 3 trial, you go to FDA or DCGI or European health authorities, and then ask them, okay, this is the data we have, and we think the drug should be approved now. We propose that drug should be approved for marketing. And then the agencies will review the entire data, and if they really think that there is a benefit to the patients and there is no safety risk, there is no concern of the side effects. I mean, there will be some side effects, but there is no serious side effect or toxicity in patients, they will allow you to go to market. But it doesn't end there. After going to market as well, there is something called phase 4 trial, which is a pharmacovigilance. So you generated data in thousands of patients in phase 3 trial, and you believe that it is safe. Once it goes to market, it will be given to millions of patients. And how do you know that the data from the thousands of patients is predicting right for the millions of patients? Because then when you go to millions of patients, different patients have different disease biology, different genetic constitution, etc. So then you still continue to follow the drug, how it is doing in the market, not from the commercial aspect, but from the safety and scientific aspect. Okay, is it working? Are the patient responding? Whether the disease is getting cured? Are there any side effects? And we have to make all these records. And that's called a pharmacovigilance or a phase 4 trial. So it continues through the life cycle of the drug. This is very, very important. And switching a little bit to non-clinical safety, that's the core topic of my talk today. Where do we play a role? Non-clinical safety plays a role right from the beginning, where you identify the target, all the way up to phase 4 trial. So different stages, we have a different role for non-clinical safety or the toxicology, but then you have to be engaged throughout the drug discovery and development cycle of the drug, and that makes it a very, very essential function or component of this entire process. Because in the beginning you will find out if this target has some theoretical risk or not. Then you will do some in vitro studies, do some in vivo toxicology studies, and then you continue for development. I will cover in my subsequent slide. Different phases of clinical trials need a different set of studies, the toxicology studies or the non-clinical safety study that you have to do. So it's a very, very important function. If you just look at